In this video I will try to explain a little bit about the web tool called the Logbook Designer. With the cellular logbook I believe that we have a nice foundation that is usable as a platform for exciting new technology, and one of the things needed to actually make the cellular logbook a system that is used by the scientific world is a quick and easy design of a cellular logbook plasmid. And that is what this tool aims to do, as well as providing an overview of the plasmids created and an algorithm that computes which sensors have come to expression after using your cellular logbook in an experiment. So the first thing that we need to do is to input the backbone sequence, which in my case is the 2012 iGEM backbone sequence. Then we need to input some sensors. There are two ways that this can be done. It can be either done manually in a FASTA or JMBank format, or we can pick some predefined sensors from the project registry database. Now I'm going to pick 11 sensors from that. And then after we press submit, the first thing that we see are a bunch of buttons. And the first button is labeled sensory plasmid JMBank 1 and so on are of course quite self-explanatory, because they create JMBank files. For each 5 sensors it creates one sensory plasmid, and this is because we don't want our plasmids to get too big. And then for each sensor it has to create a memory part that is recognized by the methyltransferase and zinc finger fusion protein expressed by the sensor, and these all go on the plasmid called the restriction plasmid. So when clicking on one of the JMBank buttons it gives the JMBank sequence in a text box. Next we see the visualization buttons labeled sensory plasmid visualization 1 and so on, and what we see upon pressing one of these buttons is a graphical display of the plasmid. Now this gives us an easy and quick overview of what our plasmid consists of. Each feature, like a methyltransferase zinc finger, is given its separate color for easy recognition, and when hovering over each feature we get some additional information such as its full name. And finally, when clicking on a feature we also get its DNA sequence down here. The final part of the tool is the readout algorithm. When many sensors are engineered on the plasmid, it becomes pretty hard to actually identify what each combination of band sizes actually means. The DNA band sizes of course being the readout data of the cellular logbook. So by just inserting the size of each band separated by a comma in this box, the web tool gives you the sensors that belong to each band. In the cellular logbook design, this means that the sensor has not come to expression because its respective restriction site has been methylated, and as such it has not been cut. So in short, this tool enhances the cellular logbook project in such a way that it makes the design of the cellular logbook plasmids a lot easier and faster, as well as giving a way to read out the results of the experiment with the cellular logbook.